Okay, well, welcome everybody to our weekly IMAG MSM working group. Today is March 11th. We have two speakers, Robert Stratford and Veronica Zarnitzyna. We're doing something new because Veronica is going to be talking to us about the work she's been doing in the subgroup. So it's the first of our subgroup reports, and I hope that will be an ongoing feature of these meetings. I remind you that the meeting is being live streamed on YouTube and is recorded and made available, so please be aware of that. And if there are any issues that come up, please feel free to contact me, Reinhardt, Jim Sluka, who's been doing the website of things, or Bruce Shapiro, who you know very well from emails. And so we very much appreciate your suggestions and feedback. As always, I want to remind you about the Slack channel. Grace Peng sent out a reminder that we should be using the Slack channel, so I'll say it again. Also, we really could use help updating and populating the IMAG MSM wiki. I understand that providing content for that wiki is somewhat tedious. On the other hand, it's a good opportunity to get your material out there. And so we'd appreciate your help. As always, we would love to have questions from people who don't ask questions before. We'll have a half an hour reserved after the main meeting for discussion with the speakers. We hope some of you will stick around and talk to them. And so that's an opportunity to ask more technical and in detailed questions. And I will provide a 5-minute warning for our speakers if it looks like things are going to run over time to keep us on time. Upcoming meetings, next week we have Yanis Kevrakidis from Johns Hopkins. We have Ashley Ford talking about modeling lung fibrosis. March 25th, Jacob has agreed to talk about the preliminary work of his subgroups. And David Otta is going to be talking to us as well. April 1st, Zhang Yin. Don't have a title yet for that one. And then April 8th, we have Denise Kirshner, who's going to be talking about her modeling of tuberculosis in a whole variety of different contexts. And also James Moore. Definitely appreciate suggestions for going forward. Anybody who's a subgroup leader who would like to present on the subgroup, we'd also very much appreciate that. Again, suggestions are appreciated. Bruce is very efficient once we have the suggestions in issuing invitations and following up on those. And they don't have to be modelers. Definitely people who could use modeling, people in public policy, funding, as I mentioned in journalism. All of these are potentially useful topics for this subgroup. And with that, I will turn it over to Robert, who is going to be talking about the role of modeling in drug discovery and development.
Great. Thank you, James. I'll go ahead and share my slides. And I've got about 15 minutes. Is that what it is? Or okay, all right, I'll jump right in. So I wanted to focus this on pharmacokinetics, although we could talk about pharmacokinetics and its link to pharmacodynamics. Because of time limitations, I'll just focus on pharmacokinetics, which is defined really by this cartoon here that you see before you. Pharmacokinetics relates dose to exposure. Exposure generally expressed as the concentration of a drug in the plasma as a function of time, although it doesn't have to be in the plasma, but that's usually our surrogate because plasma concentrations of a drug are so readily available relative to tissue concentrations. So it's our general surrogate for what's happening in tissues throughout the body. So in this cartoon here, you see before you different aspects of the exposure profile that we see on the right here. This would be an exposure profile over time. This is the integrated equation, or sorry, the integrated equation that relates concentration to dose through some different parameters. A parameter called Ka, which reflects the rate of drug absorption. If you administer a drug, let's say by the oral route of administration, Ka is the first order rate constant for absorption. And then clearance has to do with the elimination of drug from the body, Cl, and then V has to do with the extent that the drug distributes outside of the plasma and into tissues of the body. So these are three key parameters that summarize absorption, elimination, and volume of distribution, the extent that a drug leaves the systemic circulation. So we use these types of models to, there are working horses to relate drug dose to drug exposure. Models that are used in drug development really are fit for purpose. And so this entire scheme here we see before us starts from really early on when a pharmacologist identifies a biological target of interest, and then they validate it in partnership with a medicinal chemist. They develop a higher throughput screen, and they screen to identify a compound that has structural characteristics that look like the compound might be druggable, to eventually lead to a product that can be sold. Once you have a hit, you convert it into a lead, which is a more mature hit, and then you do optimization of that. And it's at this point here in lead optimization to an eventual clinical candidate selection where we start to see a lot of in vivo studies come into play here. And we rely on physiological-based models to drive the selection of an ultimate clinical candidate. There are a lot of things that happen during this lead optimization candidate selection, but models, in vivo studies and models that summarize key parameters in those in vivo studies and that allow us to forecast human exposure. Are very useful. We call these physiological-based models. And so I want to talk a bit about that. And then once we get into clinical development with a single compound, we've screened thousands, tens of thousands of compounds here, ultimately to get to one clinical candidate, we'll use another type of modeling called population-based modeling to drive decisions in clinical development in the different phases. So these models are fit for purpose, starting largely with physiological base and then ending with population based. So I just want to say a couple of things about these models in general. The way I look at modeling is that, these are models that where you have properties, drug properties, intersecting with physiological processes or biological processes. So physical chemical properties intersecting with biological properties. So this is an example. This one refers to drug distribution. It could easily be a drug absorption example or a drug elimination example or a drug excretion example. And I chose drug distribution. So we have two drugs here, propofol and its key physical chemical properties. It's pKa, which says that at physiological pH, this is largely unionized. It's got low molecular weight and it's got a decent octanol water partition coefficient. And we have two tissues here before us, brain, and this Q over V refers to the blood flow, the percent of cardiac output. Every time the heart beats, the percent of cardiac output that goes to the heart, and then that goes to the brain, and the volume of the brain. And the ratio represents perfusion. So how well brain is perfused, 0.5. And then muscle, by contrast, much less perfusion, 0.025, because of lower, both of these, larger volume and a lower proportional blood flow to muscle tissue relative to brain. So it's a 20-fold difference here. And so propofol because it's largely unionized, it is unionized at physiological pH, it's really well suited to move across the blood-brain barrier, which has a very tight, is represented by a very tight barrier, anatomical barrier, where drugs have to go through endothelial cells instead of between endothelial cells. In contrast, benzylpenicillin, also low molecular weight, a decent octanol water partition coefficient, but it's got a carboxylic acid in it, which means that it's negatively charged at physiological pH. So as a result of that, it can't move through endothelial cells by passive diffusion. And because we're talking about the brain, it can't go between adjacent endothelial cells. So it's really blocked from going into the brain because of this negative charge and the structural properties of the blood-brain barrier. So even though muscle is less perfused than brain, benzylpenicillin largely is targeted to muscle tissue as opposed to brain tissue. So just a good example of physical chemical properties intersecting with physiology. So these types of models, again, largely earlier on when we start doing lots of in vivo studies, these PBPK models serve us really well, and I'll talk about those first. And any time left, I'll talk about the POP-PK models, which are used down in clinical development. Okay, so physiological based models. So these are based on anatomy and physiology and drug properties, which is what I just talked about. So the way that these models work is that you've got an alignment here of all of the major organs and tissues in the body. And this is just a map, a schematic map of human, anatomy and physiology, where we've got blood flow, arterial blood flow coming out of the heart, going into the different tissues, and then going out of the different tissues and back to the heart and in the lung again. To be pumped back out. So you have this recirculation system, and the concentration of drug in an organ or tissue is influenced by Q, the blood flow. It's influenced by V, the volume of a tissue, and then drug transport across the endothelial barriers largely that represent, that separate the bloodstream from the interstitial water in a tissue. And drug binding and partitioning in membranes, in endothelial membranes, in tissue membranes, and the cells that make up those tissues and organs are key determinants of concentration in any of these tissues. These models are very useful for predicting concentrations of drug in target tissues. So if you have a drug you're targeting to the brain, or in the case of COVID-19, and James and I have been doing some work in the lung, with remdesivir to look at concentrations in the lung. These models are very useful for this. They have high translational fidelity. Let me talk about what I mean by that. So what I mean is that, the physical chemical properties of drugs, their size, their lipophilicity, the octanol water partition coefficient being a surrogate for lipophilicity, they're fractioned and ionized. Only the unionized form can move across. The ionized form can't move across cell membranes. These physical chemical properties, they don't change as we go from a rat and we start doing in vivo studies, preclinical in a rat or a non-rodent mammal such as a monkey or a dog. These drug properties don't change. They're constant across species. And then the other thing too is that as we move across species, the anatomy and physiology is largely, to a large extent, it's conserved. So, as I mentioned in that drug distribution example, capillaries, drugs have to move across capillaries, and we have different types of capillaries and different modes as a result of those different types of capillaries, different modes of drug movement across the different tissues. This anatomy and physiology is largely conserved as we move from a mouse, a rat, to a dog, to a non-human primate, and ultimately to a human. So these models, PBPK, physiological-based PK models, have great translational fidelity that allow us to do studies, calibrate our models in animals, and then use them to predict target concentrations in humans. So they're very useful for that. The models are continuing to develop. This is a schematic, if you will, or a cartoon of the blood-brain barrier. And as I said, drugs can't move between endothelial cells, adjacent cells, because of very well-developed tight junctions in the blood-brain barrier endothelium. So drugs can only move across by passive diffusion, provided they're like propofol and they have a nice, they're largely unionized or significantly unionized at physiological pH. If they're not, then they rely on a carrier for uptake. And the blood-brain barrier is very good about protecting the brain tissue here. So this would be the plasma here. And then on this side of the endothelial cells would be the brain. The blood-brain barrier is very good at protecting through these efflux transporters, especially on the apical plasma endothelial surface, PGP or breast cancer resistance protein. These expression of these transporters across species in mice or rats and in humans, we're starting to do proteomic analysis to be able to quantify how different interspecies differences in the expression of these transporters. So these the role of transporters and their contribution is beginning to its contribution and its ability to incorporate non-passive transport in these models is growing in accuracy as a result of proteomic analyses. So this is just looking under the hood. We have here for a blood-brain barrier target, we've got volumes in the brain for humans. We have these same volumes, and you got the, you got the whole brain volume, you got extracellular fluid, intracellular fluid, all these cerebrospinal fluid, brain microvascular volumes, you got flows and you got surface areas, and the width of the endothelial cells. And then you got your drug-specific parameters here, going down here, log P, pKa, charge, and then only the unbound fraction can move across. So we got unbound fraction. So, these properties translate very nicely because the drug doesn't change as we move from a rat to a human. And then we have these properties measured across species. So these models, do very well in their ability to translate to predict human levels. Okay, so enough said for that. So once we get into clinical development, these population or POP-PK models enable development. They create tremendous efficiencies and they're very realistic. So the way these models work is you have one or two compartment models, depending on how rapidly drugs move from plasma into tissues. For some drugs, they move more slowly, and so they're best described by a two-compartment model. And the way the concentration versus time on a log scale looks for one compartment is just a mono-exponential, but some drugs display this bi-exponential two-compartment-like characteristics. And then you can develop models for different drugs once we get into humans. And the way that we model these is in the past, 30 years ago, we did it on an individual level with rich PK sampling. We developed an individual model for each person in a clinical trial. But now with computational power, we use these population-based models. So I'll talk about those. These are known as nonlinear mixed effects models. The nonlinear phrase has to do with the relationship of concentration to time is not linear. The mixed effects means that we have a model, two models, structural model, one versus two compartment model, and then we have a statistical model that describes inter-individual variability across the population. These models are really well suited for clinical trials where we have sparse sampling because we can't get rich samples from patients when they're outpatients in a phase 2 or phase 3 trial. And we don't get times, for all of our patients at the same time. So we have unbalanced data. We have some patients with samples at 1 and 5 hours, other patients with samples at 0.5 and 10 hours. So unbalanced time points, small number of time points, not rich sampling, and these models are perfect for real, this is the way it is in phase 2 and phase 3 trials. So these models do very well. They're very good about partitioning sources of variability and identifying weights, sex, smoking, genotype, known sources of variability across a population for a given drug, and then unknown sources of variability, inter-individual, between subject variability, inter-occasion, when we administer a drug to a patient, over the course of a year, the 3 months between different doses. We can account for any potential time-dependent changes in their pharmacokinetic parameters, and then any leftover unexplained variability. So these models separate and quantify these sources of variability.
5 minutes?
Are we about time?
5 minutes.
Okay, so this is an example of some of my own work and the value of population PK. So this is looking at a metabolite, nitrite, in the plasma. So nitrate was ingested. We measured nitrate in the plasma, and then a portion of that is metabolized into nitrite. We see here the nitrite, the metabolite, over time across a variety of subjects. And you can see, quite a lot of intersubject variability. So I built a model here that accounted for nitrate conversion to nitrite. We measured both of these analytes in plasma. And this is just looking at the nitrite observed levels versus the individual model predicted levels, showing the line of identity here in solid, and then the regression line and the red dashed line. Our predictions were no worse than 2-fold off of the observed values. So, taking this population model was able to capture this inter-individual variability and quantify it and attach it to various parameters in our model, the formation clearance or the volume of distribution of nitrite or the clearance of nitrite. These models are very useful too for hypothesis generation. As you look at the literature, there are 2 different ways that nitric oxide, a metabolite of either nitrite or nitrate, can be produced in the lung and expired. And we measured these, and I'm now developing a model that has 2 different structures, 2 different pathways for nitric oxide in the lung. So these models are very good for testing different hypotheses for how nitric oxide might be produced in the lung, either directly from nitrate or through the nitrite pathway. They're also very good at, this is another example from a more recent, less recent paper where we could model, I modeled, in the literature, we had a lot of data on dopamine response, but not very much data on the exposure that generates that response. And so I used a model to simulate brain extracellular fluid concentrations of a stimulant, amphetamine, and then was able to predict, use the model to predict concentrations and draw relationships between those concentrations and a dopamine response over a variety of doses from 0.1 milligram per kilogram to 5 milligram per kilogram in rats. And then saw some clockwise hysteresis, which I then incorporated into a PK pharmacodynamic model. But simulation was a key part of this because I didn't have any exposure data at these higher doses. And I used the model to drive that exposure predictions. And then you can also use these models. I said PBPK models can be used to go from rat to humans, but these population models can too. This is a less recent publication where I used a principle known as allometric scaling to take bupropion levels in brain, here in plasma and brain of rat. These were measured values, and then was able to predict human brain concentrations relative to target potency. To make conclusions about what is the active form of the drug, either the bupropion administered or the active metabolite. These models also have different scales of representation. I talked about individual versus population. I really emphasized the population value, but we can also look at scales of representation for the whole organism, which is what I've been talking about. But these population models can be, used to, understand drug distribution in the brain in a particular tissue, especially if we sample that tissue. They can also be used at the cellular level. So this would be an in vitro study. And then we can incorporate in vitro measures into a whole body type of, so this is an integration, if you will, of very mechanistic models built from in vitro systems, and then integrated into a whole organism level model to develop. It's a very beautiful piece of work on a glyburide, hypoglycemic drug, and understanding mechanistically how this drug reduces blood glucose levels. So that's kind of that's about all I had to say. And hopefully I'll be available afterwards to answer any questions.
Great, thank you very much. I mean, it's really informative to see a set of models that are in fact used in drug development and clinical practice. So I really appreciate your being willing to give us an introduction to that important area. And I also appreciate you being willing to do it in such a short amount of time. I know it's hard to cover such a big topic in such a short amount of time. So I do appreciate it.
Yes, well, thank you. And really that was the major point that you made, is that it was largely introductory. So mostly background material and not a lot of my own research.
Okay, thank you. So now we'll move on to Veronica, who's going to talk about her work with the subgroup. This is the first of our subgroup breakout reports, and we very much appreciate Veronica's willingness to be the lead on this. And as I remind you, Robert will be available after the main meeting for conversation. We may have time for a few questions before the end of the main session, but he's generously agreed to stay on and be able to talk to people about PBPK and PK modeling and the relevance to their areas of interest. So with that, I will turn it over to Veronica. Are you
okay? So let me share the screen. Can you see my screen? Can everybody see my screen? Just not. Okay. So first, I would like to thank the organizers for inviting me to give a talk on our subgroups work in progress, and also for providing all of us with a platform where we can exchange ideas and discuss our research for bringing together great scientists with diverse expertise and experience. So our subgroup is called Innate and Adaptive Immune Response, and we focus on within host models. So we decided to meet every 2 weeks, and so far we had 3 meetings. At the first meeting, we defined the focus of the subgroup, and I will talk about it in the first part of my talk. And right after our first meeting, Reinhard and James suggested to discuss within subgroups how to build a person's digital twin. So basically how to integrate known human physiology and immunology with real-time clinical data so that we can have a predictive computer simulation for a given person. So our group, subgroup, took up the challenge, and during last 2 meetings, we had fun discussing how to start building a digital twin within host model. And I will share some thoughts from this discussion in the second part of my talk. So what I will be showing on the next slide is a product of our subgroup discussion. So I would like to right away acknowledge the contribution of people. We have colleagues from Cherry Data, Jason Schumacher, and myself. And here, actually, many more people sign up for the group, but here I'm listing people who actually came and participate actively in discussion during these 3 meetings. And we have great scientists from US, Canada, and also from South Africa. So we decided that one of the interesting questions to tackle and understand better within our expertise is very high heterogeneity in the immune response to the same viral challenge in different individuals. We know that actually some viruses like influenza almost always generate acute infection. Some other viruses generate persistent infection, like HIV or herpes viruses. But there are some viruses like hepatitis B and C that may generate both outcomes in immune competent humans. And considerable evidence has shown that the differential immune response is a major part that defines these outcomes in this and many other viral infections. So we ask a question, what features of the virus and which factors of the immune system, both innate and adaptive, affect the outcome of the viral infection? What basically contributes to the heterogeneity? And here comes also COVID, because COVID it's probably one of the best infections because it generates a huge heterogeneity in responses from asymptomatic infection to mild to really severe. And also during the last year, lots of data has been collected and also made publicly available. So we ask a question, is it possible that SARS-CoV-2 actually establishes a persistent infection in a fraction of otherwise healthy individuals, at least at the moment when they're infected? And are long hauler dealing with organ damage after virus clearance or with low-grade reactivation of the virus? And long haulers, were first reported in Italy when people, even after a few weeks of COVID, are still showing the COVID symptoms. And so basically, long haulers could be defined as people who had a confirmed medical diagnosis of COVID, but then after several months, they still did not return to pre-COVID kind of health, level of health. And they are having symptoms that suggest long COVID, but often no evidence of permanent damage to the lungs or heart or kidneys that could cause those symptoms. So next slide is showing some data. And this is viral load kinetics and also seroconversion and clinical observation in individuals. I listed 6 out of 9 reported individuals here just so that the panels were bigger and easy to see. And you could see the viral load was measured in throat swab, yellow color, also in sputum, orange color, and in stool gray. And what you can see, so the question is how do we define acute infection and persistent infection? If we define acute infection as infections that would be cleared in like 2 weeks, then looking at this data, you could see that like even majority of these people would pass the kind of definition of acute infection. And this data you might already see before because they were in Alan Perelson work. They actually did some modeling to fit this data. And you could see that if you look at the sputum, this is basically show you infection in the lung. Then like in the top left, there will be a spike after a few weeks. And in some other people also, you could see some spikes. And what they did is they use extended target cell limitation model, trying to mimic some additional kind of infection in the lung, as if like lung infected in patches. So they introduced additional target cells and feed this data. So another data also, the previous was on Nature, and this data also in Nature from a Nussensweg lab. And it shows a immunofluorescence imaging of intestinal biopsies taken 3 months after onset of COVID symptoms. And region in white boxes enlarged on the right. So, and arrows, white arrows, they indicate enterocytes with detectable SARS-CoV-2 antigen. Also, this paper reported persistence of SARS-CoV-2 nucleic acid and immunoreactivity in the small ball of 7 out of 14 individuals after 4 months, and all people were asymptomatic. So at least it's look like in some people we can see the persistence of the antigen, and in those people also immune responses consistent with persistence of the antigen. Also, I found a few quite alarming reports about long COVID in children. One appeared very recently on medRxiv, and they just took in one hospital 129 children diagnosed with COVID, and then 120 days later they look at those kids, And very high percent of kids still have at least one symptom, and many of them have 2 or 3 symptoms. And of course, symptoms correlated. If you have a severe infection at the beginning, then you had a higher chance to still have symptoms. But what's surprising that some kids were asymptomatic and still have later on some symptoms that could be related to COVID, like shortness of breath and like other typical COVID symptoms. So group action plan. So we, of course, will review available data. Then we will make an inventory of the models able to capture both acute and persistent infections. Then we will build a set of alternative models and bring them in contact with either SARS-CoV-2 data or other viruses data that we will find and that could be useful. And also my colleague suggested to add the discuss finding strategies here as well. And usually when you think about modeling, if you want to model acute versus persistent infection, you might think that a large number of different factors from the virus, from the different immune cells, cytokines, like lots of stuff may contribute. But recently, and we discussed it with our subgroup, one paper was published in PNAS 2 years ago, and it used a very simple model. It's only 2 equations. And this model beautifully capture both like persistence versus clearance and also the exhaustion mechanism. And so basically you see 2 equations. One is I stay for the infected cells, and the growth of infected cells has been modeled using a logistic term with the per capita proliferation rate K1 and also carrying capacity Imax. And infected cells were killed by CD8 T-cell immune response. So it's this term. And CD8 T-cell immune response, the activation, it was 2 terms. One for the activation of the immune response and one related to the T-cell exhaustion. And both of them were modeled as a Hill function, and the model parameters were set up so that K3 was less than K4 and Kp was less than Ke, and that allowed at a small level of antigen to generate CD8 T-cell response that contribute to killing, but at a high level of antigen, it's actually put the CD8 T-cell response into exhaustion state. And then on the top, you could see that the model on the top right, you could see that the model has 3 steady states, and it has 2 stable steady state, one for clearance and one for persistent, and also one unstable saddle point that separates them. So it's like relatively simple model, but it captures the main features of acute versus persistent infection. Also, what you could see in the middle row, they plotted the individual trajectory for infected cells, for CD8 T cells, and also for cytokine pathology. Pathology was defined by the equation I show on the left. It's proportional to both infected cells and CD8 T cell response, and the decay with a linear term. Somehow it's going back. And what they show that actually the maximum pathology happens somewhere in the intermediate range. And it's all very nicely kind of describe what happened in the reality with this very simple model. So what we will do, we will still keep reviewing other models in the field to find a really nice templates. And then we will, of course, add many more. Actually, even this paper, they added, for example, influence of NK cells, influence of the viral inoculum, influence of the innate immune response. They modify their model by adding more equations. So we will do something probably similar. We will see how it goes and what other models we will find. So Now, let me switch to the digital twin challenge in the second part of my talk. So at present, we cannot accurately predict either short or long term how the immune system of a particular person will react to different viruses. And this is true not only in the case of new pandemic virus, but also for circulating viruses, including seasonal influenza viruses. We just simply could not predict for individual. All the available clinical information allow us to empirically characterize responses in a population, but when it's come to just a given person, our kind of ability to predict is very limited. And this, of course, is very critical if you want to make a prediction, if you want to make treatment, and also if you want to answer some questions like what determines severity of disease in similar people, similar, for example, of the same age or of the same gender, or if you want to suggest how long the vaccination lasts and how often elderly will need to get a booster coronavirus vaccine. So it would be nice if we have this digital twin for each person and can simulate the intervention first. So we ask a question, what are the main gaps that prevent predictive understanding of the immune response, and how can we start filling those gaps? So here, if you want to build a digital twin for a person, we will need to take into account at least 3 major components that collectively drive the immune response. And you could see here, it's a person's genetic makeup, and it include HLA alleles and single nucleotide polymorphism. Actually, there are striking examples. For example, it was shown that certain HLA-B alleles were found to be prevalent in so-called elite controllers in HIV. So people maintain an undetectable viral load, even in the absence of previous retroviral therapy. Also, it's important to characterize the immune status of a given person, and this will include, it's like personal, basically CBC, cell blood count, and also the history of all infection and vaccination, because each infection, each vaccination will change the repertoire of T cells, B cells, antibodies. So it would be nice if we can access it somehow. And also, we would like to access the physiological status of the person, and that will include, of course, age, cardiovascular health, microbiome, level of electrolytes, vitamin level, stress level, like everything here. Then
about 5 minutes, if you can.
Yeah, okay. So then how do we deal with it? So if you want to model a person, then we can even come to it as a black box, but then we need to know the baseline of this black box. And we also need to have at least several examples of the immune response for this person. So one of the idea was, what if we use individual responses to seasonal virus as a stress test for the immune system? And seasonal virus, it's usually influenza or seasonal coronaviruses. And here on the left, I show the graph from Kucharski paper, and they estimated how frequently people infected with influenza through their life. So they use the cross-sectional antibody titers to 9 different strains spanning 40 years of circulation of influenza A, H3, and 2, and together with mathematical models to do the estimation. And although infections became less frequent when individual progressed through the childhood, you could see that they occur at roughly similar rate after 40 years of age. And each person will be on average infected with H3 and 2 subtype maybe once in 5 or 7 years. And there is another subtype, and then there are pandemics. And for seasonal coronavirus also, you can find some papers. This is more on a very frequent side, because it says that every 12 months, and we have 4 coronaviruses. We also have now SARS-CoV-2. Basically, the idea is, if you take, for example, 100 people, characterize them, and start watching them for a year. During this year, they will be infected with these seasonal viruses at least once or twice. And we know how to, what are those viruses, and we can detect them with a blood test, just looking at the boost of the antibodies. Ideally, we would like to have test every like 3 or 4 months. So the question is then, what data do we need to predict individuals' immune response to viruses? What data is available now? What level models can we use? So, and our suggestion is just to use these coronaviruses and influenza viruses as a stress test for the immune system and start to collect this data. Also, Amber Smith actually pointed to a very nice study that was done in Stratford group. And this is a like potentially a very nice opportunity how to proceed with this digital twin project. So what people did here, they used retrospectively smartwatch data. And here you could see they monitor people, and some of those people were known and detected to have COVID. Some of them were detected to have some other illness. Also, some people were sick but without diagnosis, and some people were not sick but had a high rate of exposure. And what they did in here, they use the smartwatches to track heart rate, steps, and sleep. And what they found that there are several predictors. And what is very exciting that those predictors, at least in case of the coronaviruses, they could detect that the infection is coming and the disease is coming well before the symptoms. So in some cases, it's like 9 days before the symptoms. Actually, they say that in 63% of the COVID cases, they detected before the symptom onset. So that could be a way to go. Even because there are some studies that already collected, like for example, swabs for influenza. And if those people were wearing the smart devices, we can retrospectively go back with the idea of extracting from those data for example, some correlation between the viral load and those parameters that estimated in this paper. Because as soon as we have a viral load and we have responses, antibody and T cell responses, we already have lots of models to start play with, or already kind of a big pool of developed models. And here it's again, it's a heart rate. So how it's raised above the baseline level. And also they found useful another parameter is a heart rate kind of divided by the steps. So people start to walk less when, even if the infection did not show up as the symptoms yet, people already behave differently. And then if we get the data from people, we have lots of models within host model already developed. And this is a very nice figure from Amber Smith's paper. And it shows a viral dynamic. This is a target cell limitation model with eclipse phase. It shows innate dynamics. It shows adaptive dynamics. And people already analyze how the viral load in the person looks through the time. So we can use all these kind of preset models, and here I listed some other models and equations. And basically that's where we are with our subgroup. So I put again the slide with people who contributed to discussion and to all these ideas. Okay.
Thank you very much. It's impressive output for 3 meetings.
Oh, we have a great people. If you look at the names, great people. We are lucky.
Okay, thank you. So Reinhard, is there any business you want to go over today before we have questions?
No, as usual, I don't, but let me just thank both speakers. It was very, very interesting presentations. And I think what Veronica showed is an example of what the different subgroups are working on in you know, some focused area of this whole larger problematic. And we've had some very interesting discussions in the steering group as well. And as a reminder, the steering group consists of all the co-leads of the subgroups. And we should pretty soon be at a point where we can put together some sort of a document that everybody has access to and can take a look. We've had quite a few discussions about, you know, what are some possible outputs. So Veronica talked about the digital twin. That's certainly one direction one can go in, you know, personalized predictive models of disease. But of course, there are other possible endpoints of where one could go is comprehensive models that one might consider essentially as a research tool that might help both computational and experimental research. So there are many different directions one can go in. I think the steering group discussions have been extremely insightful. So if you've not signed up for one of the subgroups yet, please do. It's going to be very interesting. And I think, you know, one of the deliverables for each of the subgroups is a white paper of some sort, which could turn into a publication. And I think Veronica, can you just say a little bit whether your subgroup has any ideas of what you might want to do with putting this work together in some form?
I think we can write some reviews probably after we review the models for acute versus persistent. That probably would be one of the options. I don't think we can so quickly do some like lots of research.
I think laying out a direction of research is already very valuable. So I don't think the white paper has to be a product in the sense of finished research, but I think you've already enunciated a very nice approach. And I think on Robert's side, he was talking about a very well-developed set of modeling approaches, but it could be interesting to think about whether there are specifically viral infection and immune-related questions that would be relevant in the context of PBPK and other models. In other words, is there, in Robert's case, it looks like you have very good data, you have volumes, you have partition coefficients, you have very complete data sets in many respects. But it would be interesting to ask the question, are there pieces of missing information that would be specifically related to viral infection or immune response that could make those PBPK and other models that you described more useful?
Right.
So,
yeah, this is something we'll be discussing in our subgroup.
So I think then, I guess we're pretty much out of time for the main meeting. I really hope people will stick around and speak to our speakers today, because I think they were both exceptionally interesting introductions. If you can't stick around and talk with our speakers today, I hope you'll contact them individually. Reinhard, is there anything else then to cover?
No, thank you. I just want to thank Ruchira for putting all the links to all the papers that that people mentioned in the chat. That's very helpful. Thank you, Ruchira.